Right, I think we'll get started. So welcome everybody to the fourth and final webinar in our Centre for Forest Protection webinar series that we've been having over the last couple of weeks, which are reporting on our projects in phase 1 of the Centre for Forest Protection research. So my name's Louise Gathercole. I'm the coordinator for the Centre for Forest Protection. So I work with the scientists and with our funders at DEFRA. I've arranged these webinars for our scientists to have the opportunity to speak to you about their very recent results. So we have 3 projects for you today. They're all sort of roughly around the same themes of research that's being done using tree breeding or genetic methods to predict risk and understand what the risks might be for some of our more economically important tree species. And for those of you who aren't familiar with the Centre for Forest Protection, it's a research collaboration which is led by Forest Research and Kew and funded by DEFRA. In the first instance, we were funded by DEFRA Plant Health, and we're working with Plant Health and the Forests and Trees team for phase 2, which will start in April. So the focus of the centre is really very much on the future of forests and future resilience, looking at the various different kinds of environmental and socioeconomic threats, and using innovative science and interdisciplinary collaborative research to help inform stakeholders like many of you. If you want to know more about the centre, we have our own website, and there are information about all of the projects that are being presented today on there. If you haven't signed up via signing up for this webinar, you can also sign up for our newsletter, so that if there are papers being published or we have a blog or new projects, you would be able to find out more from our newsletter. So without further ado, I am going to hand over to Jaz Stoddart, who is a postdoctoral researcher at Kew, who's going to be talking to us about his project modelling potential risks to non-native tree species from UK native organisms. Over to you, Jaz.
Thank you, Louise. Good morning. Like Louise said, I'm Jaz. I'll be discussing our efforts to predict the risks to non-native tree species in the UK from native pests and pathogens. As I'm sure many of you already know, introducing non-native trees is expected to be one of the ways that we're going to strengthen our forestry sector going forwards. Non-natives can provide a range of benefits, such as higher productivity, higher value timber, greater resilience in the face of a changing climate. However, these benefits are not without some level of uncertainty that they bring. So the risk here is exemplified in this cautionary tale from Sweden. They embarked on widespread planting of lodgepole pine in regions in which a local fungal pathogen responsible for bronchiostid disease was found to be hosted by the lodgepole pine quite damagingly, in a way that the local Scots pine populations were not similarly affected. The result was significant economic damage and significant losses of the plantations. So the question now becomes, if we had known that this interaction was going to happen beforehand, could we have prevented it? If we had been able to predict it? We believe we could. So if we had been able to predict it, this interaction could have allowed for informed decision making at a stakeholder level. Knowing that such an interaction was a risk efforts could have been made to identify the severity of that risk through planting trials or inoculation of some trees with the fungus. And then upon identifying how severe the damage could be, options could have been explored to avoid the sort of economic impact that would have been associated with the plantations failing on that scale. Alternate tree species could have been selected, or planting could have been restricted to regions with a very low prevalence of the fungi. So the aim of this project was quite simply to find a way to make such predictions a reality, so that we can hopefully prevent this happening here in the UK and sort of anywhere else in the future. Our work aimed to address the uncertainties that are associated with introducing non-native tree species by developing a methodology to estimate the likelihood of novel interactions between non-native trees and the native pests and pathogens found, in our example, in the UK. We then sought to demonstrate how these predictions could be used to identify the interactions that are most likely and therefore probably warrant the most further investigation, with the hope being that this methodology could then be used as part of a greater decision support framework for stakeholders. So before I go on, in our sort of proof of concept work that was embarked on in this project, we selected 28 non-native tree species that have been identified as potential forestry candidates for the UK. They're listed on this slide, as you can see. We have an even mix of conifers and broadleaves with a range of genera represented. You've got firs, maples, alders, cedars, eucalypts, southern beech, spruce, pine, redwood, and then also red oak, western hemlock, and the tulip tree. It's a pretty diverse spread, and hopefully this means that our methodology is widely applicable. So onto the approach. We developed a methodology which is totally novel in how it combines phylogenetic information, so the tree relatedness, with the similarity of the native host ranges of the threats that are present on these trees. And then we hope to use this to give us a predictive framework which is able to predict the likelihood of interaction between any combination of species in the phylogenetic tree and the sort of matrix of threats. The rationale for modelling in this method is that phylogenetic relatedness between native and non-native host species is a critical predictor of host threat interactions. This is a fact that's already been shown by a number of other studies. Of course, the logical basis for this is, as illustrated in the diagram, pests and pathogens are more likely to exploit those non-native trees which are more closely related to the native host species. And this is, of course, because they are more likely to share the traits that make them suitable hosts. An example of this is, of course, the emerald ash borer affects a wide range of ash trees, from white ash, green ash, black ash, blue ash, despite the fact that these are closely related tree species, but not necessarily found geographically in the same areas. But they all share the same traits that make them suitable hosts. Now, the other part of the approach, of course, is the sort of host range similarity, and the rationale for using this data in our models is that threats with similar host ranges are likely to have similar host ranges among novel hosts. So if the hosts in the UK that they are normally on in their native range are, if they have a lot of overlap between two threats, then they are expected to share that overlap when we introduce potential novel hosts. This is because if a pest or pathogen leverages the same vulnerabilities, then they will continue to leverage those same vulnerabilities if they're identified in new novel hosts. This diagram sort of illustrates that you have very similar ranges in sort of the vertical distributions of some threats. And this is notable because green here is fungi and blue are insects, and you can actually see quite a lot of similarity between these two threats here, despite the fact those are Heterobasidion anosum or conifer root rot, and then Xyloborus dispar or the European shothole borer. And so despite being, totally unrelated threats and species, they are having largely overlapping host ranges. And this means that compared to other things, they're more likely to predict the interactions that each other have with threats. And this is, of course, important for the modelling. So to get a big picture perspective, we sought to use a broad data set. It was over 600 threats and included insects, bacteria, fungi, oomycetes, and viruses. Now, selecting a data set of this size wasn't without consequence. It was such a wide range of threats that it was impossible to model them all together, because the chosen method of statistical analysis, a multivariate Bayesian statistical regression, if anyone's interested, is computationally very demanding and does not lend itself well to parallelization, meaning no matter how powerful a computer you run it on, it's not going to speed up particularly. So you might think this is a problem, and it would be. However, if you think back to the previous slide, for any given single threat, the majority of other threats are not actually going to be providing anything useful in predicting interactions, as they don't share a lot of similarity in their host ranges. Therefore, we found we only need to consider the threats that contribute the most information to modelling each threat. And this led us on to refining the approach so that the input data was targeted to focus on the threats with the greatest similarity to those of sort of the focal threat. This resulted in us running multiple models, essentially a targeted model for every focal threat, or for every threat in the matrix, but this was still much faster and much less computationally demanding than trying to run them all at once. And then for each model, a cluster of similar threats was created and used as the input. So that meant that based on the statistical similarity of the host ranges of very disparate threats, and while removing redundancy, we were able to create small groups of threats that provided significant predictive power, and essentially just removed all of that bloat that comes with including data which doesn't contribute to the modelling. And so now the question is, did our refined approach make good predictions? Yes, we think so. We performed validation tests by withholding a number of known interactions. This is for non-native trees that we already had some knowledge of interactions with pests and pathogens found in the UK. We excluded those from the input dataset and then validated the likelihood of those predictions against the fact that we already knew that they were likely. And we were pleased to see that the model correctly assigned notably higher probabilities to the withheld known interactions, and did so in a statistically significant way. On the slide, here we have a figure of a survival function plot. This shows how many interactions for the withheld known and the other unknown interactions remain above various cutoff thresholds of likelihood. What it visually illustrates is the model's ability to differentiate between the known and unknown interactions, and how at a much larger proportion of those known interactions that we were using for validation remain at the higher likelihoods as the threshold increases. So as the threshold goes up, we've still got a lot of these known interactions, whereas the unknown interactions have dropped off significantly. This suggests that we are very accurately predicting that known interactions are more likely, while not including the vast majority of other interactions, which, because this is a matrix of all possible interactions of threats and hosts, most interactions are not going to happen and are very unlikely. So now that we know that the model is reliably returning those accurate predictions, the question becomes how to identify which interactions are of interest to prevent more situations like the one in our cautionary tale at the start. If you look at this survival plot, it's actually the same information as the previous plot, but here presented in numeric form on a logarithmic scale rather than as a proportion. And you can see that we started with just over 17,000 possible interactions for the unknown interactions. And this is a huge number among which we will find potential novel interactions of interest. But there's too many interactions to, if we wanted to assess each one for severity, things like that, 17,000, a lot of legwork, too much. So to reduce the number of interactions, we can set a minimum threshold for likelihood, and that threshold can then be changed to be more or less stringent, reflecting how many interactions we want to capture and the level of risk aversion that the stakeholder may have. So in this example, I've set thresholds at 0.1 and 0.3, and at 0.1, the number of interactions for the unknown, which is where the novel interactions are, has fallen to around 500. I realise on a logarithmic scale it's a little unclear, but trust me, it's about 500, and that means we've already removed 97% of the interactions, leaving us with just sort of the most likely 3% in this area here. However, 500 is still quite a lot of interactions to consider. So if we were to be a little bit more stringent and select threats that exceeded the threshold likelihood, or interactions that exceeded the threshold likelihood of 0.3, we're left with only 50 interactions, the 50 most likely interactions. And this allows us to focus on these most probable interactions and reduce the chance of false positives, providing sort of a far more compact list at the risk of potentially filtering out some real interactions that were predicted with lower likelihood, but with the added benefit of removing a lot of very unlikely interactions that would just be a waste of time to investigate. So, using sort of the aforementioned methodologies of setting thresholds, generating interaction likelihoods, and then setting thresholds, stakeholders can directly take that list of interactions yielded from thresholding and use that to inform whatever they want to do next, whether it's species selection to identify, for example, just which non-native species potentially have the fewest risky interactions, assess the likelihood, or not likelihood, sorry, the severity those interactions may have through trials, things like that, or even direct breeding if a non-native tree species is of such great interest that it must be had. Perhaps breeding and selection of resistant varieties then becomes an option, but only because we are informed of the risk that is associated with the interactions before we embark in the planting. So to conclude, we developed a novel framework that contributes a sort of robust data-driven method for evaluating the risk that native pests and pathogens may pose to non-native tree species. And it harnesses only very readily available host range information, phylogenetic relationships, and then through using sort of customizable thresholding, it delivers quite actionable probabilities and lists of high value or high risk interactions that may be worth or may warrant further investigation. And then these can hopefully be integrated into a broader forestry decision-making framework. So that's sort of the project. I'd like to acknowledge that I completed this work alongside my supervisors, Laura and Richard, and we'd like to extend our thanks to the many people who took their time to offer us insights along the way. And thank you for listening. We also did have a specific sort of point of interest, which is if you'd like to access this work's findings and have thoughts about what format you might like to access them in, we would like to hear about that. And so please feel free to contact Laura at the email at the bottom of the slide. So that's lkelly or l.kelly@q.org. Thank you.
Fantastic. Thank you, Jaz. Really interesting. And it sounds as if you had to go through quite a few stages of trying to work out how to optimize the model to come up with what works best. I've put Laura's email address in the chat as well. And I know that Laura would love to hear your thoughts about the best way to use the work that she and Jaz have been doing. So I'm going to hand over to Rubén now, who is one of our researchers at Forest Research. And he and Thomas Baer, who works with him in wood properties, are going to be talking about their project on Sitka spruce. Rubén, over to you.
Thank you. Yeah, thank you, Louise. Yeah, thank you everybody for being there today. Yeah, so Thomas and I were going to be talking about this project we have been developing and delivering during the last 3 years. So yeah, I titled this presentation as that because it's I think more appealing than the title of the project itself. So this is about using available genetic resources to build economic resilience, and everything is based on Sitka spruce. We will discuss how this can be also be transferred to other species. So before I get into any detail, I want to introduce the team. Most of this project has been delivered by Forest Research, mainly 2 teams involved: the tree and wood properties team and the tree breeding team. So the core team for the project has been actually something across FR teams. So with Richard Whitted and myself co-leading the project, and then Thomas Baer and Domen Fingar basically doing the hard work for us. We have been supported also by people abroad, mostly from INRA in France. That's a lab of forestry economics. And then we have had also contractors have helped in specific areas, also from INRA and from the Slovenian Forestry Institute. A very big thanks to everybody involved for their hard work in these 3 years. Otherwise, this would haven't been possible. So a little bit of background of what the situation is and why we did what we did. Well, this is something everybody knows, right? Sitka spruce is the main commercial species in Britain with 60% of the timber production. Now, as you also probably know, there has been a breeding program run by Forest Research since the 1950s and has been very successful in delivering notable genetic gains in terms of volume, growth, and yield. And that's what the result of that is that we typically called improved Sitka spruce. Of course, there is many types of improvement, but the improved Sitka spruce we are discussing here is in a way the premium one, the best improvement we can have, the full SIP. So to do this, the breeding program has established incredibly large infrastructure. So basically, we can say that we know a lot, really a lot, about the genetics of at least one provenance of Sitka spruce. Another topic of interest, of course, is climate change. So we know that drought is expected to be more common in here in Britain in the near future. So as a result of all this, we could safely say that we have 60% of the eggs in the same basket, or 60% of the timber production in the same basket. And of course, this is a little bit scary, and everybody knows that, and things have been done in order to deal with it. But what we did in particular was to formulate the following research question: can we breed for drought tolerance using the genetic pool of improved Sitka spruce? So what we know about Sitka spruce through the breeding program. And mind you, this is slightly different from what other people are doing in terms of genetics or quantitative genetics anyway. So often we see approaches where people try to select among provenances, which is of course a very valid approach, or of course selecting among the species. But here we're actually focusing on selecting among the families of one provenance, which is the formerly known QCI, nowadays Hathaway. And the reason we want to do this is because it's a very easy win if you think of it. We know so much about the genetics of this provenance that actually with very little effort we can actually try to find out if there is drought resilient that is heritable in here. So in order to do all this, we developed or organized the project in 4 work areas. The first one is the core for the project, right? It's the genetic control of relevant traits. A trait is something that you can measure, and that informs of something. So in this case, we want to find something that is measurable and that informs drought tolerance. So which trait informs resilience in Sitka spruce? And then we want to know whether the trait is heritable or not, because if it's not heritable, then we cannot select for this favorable trait. The fact that a trait is measurable doesn't mean that it's easy to measure, and sometimes it's not. So in order to make it possible to deploy the breeding, this kind of selection in the breeding program, then you have to make sure that you have something that relates to that trait and you can measure easily. So that's what we try to do in the work area 2, study of resilience proxies. Yeah, so this is mostly about how do you do this operationally. The third work area was about economic quantification. The main question here was, okay, we can select for very drought tolerant families, but how does that play out in terms of growth, for example? Is there any trade-off? That's the kind of question we wanted to answer. And then finally, there's a work area on knowledge exchange, of which this event is an example. And thank you very much, Louise, for organizing it. In this talk, we are going to focus on these 2 work areas: the genetic control of relevant traits and then the economic quantification. The first one is going to be delivered by Thomas, who has been the main contributor in it and also in the whole project. So, and before I hand it over to Thomas, I want to say as well that this has been the project has been possible because of the very, very large infrastructure that forest research has, and the effort and dedication of generations of scientists in the organization, as well as long-term funding to do these things. So over to you, Thomas. The ground's yours.
Thank you, Rubén. So I'll give you an overview of how we basically went about determining possible genetic control of drought tolerance in Sitka spruce and what we found. So we used timber samples from 2 sites in the UK. So one located in the north called Spade Adam in North England, and the other one was in Wales called Radnor Forest. Now we had samples stemming from almost 600 trees belonging to 29 different full sibling Sitka spruce families. And we have the genetic history, the genetic ancestry of these families. So basically the genetic parents are known. And we also have the same families present on both sides, which allows us to run comparisons and calculate correlations. Now I'd like to give you a quick description of how we got from the tree to the tree ring level analysis of growth and density. So we start by taking a cross-section from a cross-section disk, basically from each tree. We can see this here in this photograph, and from that we take a central section, cut out a central section, and this gives us basically the whole cross-section of the tree, basically from pith to bark, showing all the rings. And from this we then cut a 2 millimeter thin sample, and this we can then X-ray scan using an iTrax machine that we can see here in the right-hand corner. Now these X-ray images, as we can see an example of below here, were then analyzed using tree ring analysis software to measure both the ring width and also the ring density. The ring density we can see here in the different shadings. Basically, the darker late wood is more dense and the lighter early wood is less dense. And after cross-dating then all this data, we can confidently assign the growth year to each ring. Sorry, the animations got a bit messed up there. So the resulting ring widths are shown here. So each line represents one tree for each site, and basically showing how many millimeters each tree grew for each year. Now these curves need to be detrended, which essentially means removing the age and also the size and competition effect from these curves. And then leaving us with a clearer picture of the effect that climate had on the tree growth. Now, the approach we used for detrending was by fitting radial increment models for each site. Now, these models then allow us to calculate the predicted ring widths, shown here in red. And by dividing the measured ring width and the black curves by the predicted ring widths in red, we then are left with the more smooth detrended ring width, and they highlight the effect the climate had on ring growth. Now, these detrended ring widths we then use to assess the tree's reaction to drought events, basically meaning how much did each tree grow then during, before, and after a drought event. And for this, we used 4 different indexes. So how we chose the drought event, I will show you in a moment, but the drought years we were working with are 2003 to 2006 for one of our sites. All the indexes are calculated using the pre and post drought growth, as well as the growth during the drought event. So we calculated the pre and post drought growth by taking the mean of the ring width from the 3 years before and after the event. And the growth during the drought event is just the mean of all the ring widths which fall within the drought event of interest. And the first index we used was resistance, which is an indicator of the tree's ability to resist the drought event, basically looking at how much did the growth decrease during the drought event of interest. So we compared pre-empt basically the during drought event growth. The second index is recovery. This indicates the tree's ability to recover from a drought event by comparing the growth after to the growth during the event. And the last 2 indexes are resilience-based indexes, basically shows the tree's capacity to withstand a drought event in terms of growth, basically how quickly does it bounce back to pre-drought level growth rates. And for this, you compare the pre and post-drought growth. And in the case of relative resilience, you also include the growth during the drought event. So to identify the drought years, we used a combination of 2 approaches. So one was carbon isotope ratios. So during a drought event, a preference of basically which carbon isotope is favored by a tree during photosynthesis shifts slightly. So an increase in this ratio can be an indicator of drought. The other approach we used was using climate data. So specifically potential evapotranspiration and precipitation. And this is used to calculate something called the standardized precipitation and evapotranspiration index. So basically, we see that here in the bottom graph, more negative values indicate more severe drought conditions. So it's basically an indicator of a water balance. So in this case, this was for the site in North England, and we found the years 2003 and 2006 based on the 2 approaches were highlighted as drought events, and we in this case focused on the 2003 event. Earlier drought events we could not use because the trees either were too young, or depending on the size of the tree, you might not have enough tree rings in the chronology to calculate the pre-drought growth. So now what we were interested in was whether we can see any evidence of genetic control of drought tolerance using the previously described indexes, or call them traits. Now, indicators of genetic control could be when we see that different families show significant differences on average to each other in their growth response to drought. Another indicator would be if the same families tend to show the same response on both sides. So our statistical analysis showed that some families did show significant differences in their resilience and resistance to the baseline. So here we can see how many families on each side showed statistically significant differences to each other. So, for example, for resilience, 11 out of 32 showed significant differences. So if we maybe look at an index a bit closer, for example, recovery that showed no difference between families on average, and resilience, which showed some indication of significant differences. Here on the right, we see recovery. The averages of each family, so each box represents one family, is very close to the overall average indicated in red. So we saw no significant difference between families for the index recovery. However, for resilience, a certain amount of families did show significant differences. Something else we see straight away here is that there is a lot of variability also within each family. So each family does have quite a variable reaction in terms of growth to drought. Now, another way to see whether there's genetic control of drought tolerance would be to see whether the same family on one side will show similar growth reactions to drought on the other side. Meaning, are the more resilient families at Spade Adam also the more resilient families at Radnor? So, sorry, I don't know why my animations are going a bit wrong here. So here we found that only resilience showed a significant positive correlation between the two sides. This means that a more resilient family on one side also has a moderate tendency to be more resilient on the other. Now, this could be interpreted as a degree of genetic control of trees' resilience to drought in terms of growth as measured by this index. We do not see significant correlations for the other indexes, although they all tend to be positive, but unfortunately they were not statistically significant. So we can see that, especially for resilience, we can see that there is a slight correlation between the two sides. So we did see a lot of variability between sites, but also within families, and this does suggest that there is a strong interaction between site, or call it environment, and the genetics. With this, I'll hand back to Rubén.
Okay, thank you, Thomas. So essentially in this part, well, as Thomas already said, but as just to summarize, if we use these indexes as our trait, the traits that we're trying to identify, as to inform drought resilience, what we see, the first thing we see is that, as Thomas said, there is a lot of genetic variability in the breeding population of Sitka. So this is actually very good news, because we have been selecting quite a lot for growth, and still we have all this variability left, and that's very important. Then, as Thomas said as well, these traits are heritable to an extent, but there is also this very strong interaction between the environment and the genetics. So to answer our question, yeah, whether we can breed for drought tolerance using what we know of improved spruce. The answer is yes, we could try with this we observed. However, given this strong interaction between the genetics and the environment, we believe that we are not in a position to where we have all the evidence needed to really go for it and say, okay, pick these families and then select for them, because you could very well take these families and select for them, plant them somewhere else, and then they're not working because of this environmental effect. So our recommendation is for now to keep all the variability you can, and then as more evidence is gathered, then we can move forward with it. So the other part, the second part of the presentation, is about the economic quantification. And initially, as I said before, our idea was to focus on these more resilient families. So the idea was to simulate the profitability on the most resilient families under different climatic scenarios, different management alternatives in 4 different sites, and then compare to the other families, and then to see whether there was a trade-off between looking into resilience and then profitability, in other words, growth. However, as I said, we cannot really recommend to select for any specific families. So we changed a little bit this, and we try to do something that is still informative and useful, but with quite a different angle. So what we would try to do is to simulate the profitability of improved Sitka spruce, at least the improved Sitka spruce that is present in those experiments, all these families, under all these scenarios. And then that in itself is quite interesting, because we are focusing on climate change and what will happen in the future, but then compare also with the non-improved Sitka spruce and see what happens with non-improved Sitka spruce also in the future, and whether it's relevant to keep it or not. In order to do any form of economic quantification in forestry, you typically need a growth model. You need a growth model in order to project how your trees are going to grow into the future, and think that as growth as just the same, or almost the same as income. So that's why you need a growth model. So we didn't have a growth model for improved Sitka spruce, so we put together something that comes from our experiments. Please don't get excited, because I know that everybody wanted to see a growth model for improved Sitka spruce. This is based on a very limited sites. So this will be developed in the near future in collaboration with our colleagues of mensuration. But for the sake of the example, this is very valid in here. So just to show you how this works. So this graph mostly shows how the simulator in a schematic way, the growth simulator. So what you need to give the simulator is first a list of trees. So in this case, we picked 4 sites. These are genetic experiments from Sitka. We provide the simulator with the diameter distribution of these 4 sites at a young age. So these are young trees that are ready to grow. Then we pick 3 climatic scenarios, and the present scenario, current climatic scenario, then a mild emission scenario, and then a severe scenario. And then we give that to the growth model. So this is the growth module, and then so what it does is it picks these trees, and then they grow them. They grow them for 1 year, and it calculates the volume, how much volume is in there. This is passed into the economic module, and then the economic module calculates the profitability at that point, right? We need to iterate that over time until you get to the age you want to get. So in this case, our simulation ran up to 40 years, I believe. And at the end of the simulation, what you have is your projected volume for every family, for in every site, as you can see in this graph. We also have a wood properties module. I'm not going to get into this today, but in the end, what I'm going to get into today is this final thing, is you get a projection of profitability. Icing on the cake, we repeat the whole process many times, in this case 100 realization. And this is because this is a stochastic simulation. This means that we are taking into consideration all the errors of all these models. All the models have errors by definition, and that inform about the uncertainty of a process. So it's very important that we capture this uncertainty so that we can tell managers or stakeholders in general, your well, your profitability is this in average, but it could also be this, and it could be also be this with this probability. So that's also quite interesting. So we ran 100 simulations in here of realizations with over 60 million, well, about 60 million ring projections. I'm going to touch a little bit on this. So that's the profitability, the idea of profitability. So we used two different metrics to account for this. One was the net present value, NPV, and then the other one was the land expected value, LEV. I'm not going to get into any detail of how this works, but just believe me, this implies this is profitability. So in this graph, you have our 4 sites, the 4 sites that we used for simulation. On the y-axis, you have profitability, LEV and NPV. The blue line is the LEV. Let's talk about LEV only for the sake of the example. And here you have on the x-axis, you have H. You can see it, what this implies is that basically at the beginning you are in negative because you made an investment, but your trees are very small, so you cannot really sell them. At some point, your trees are big enough that if you sold them, you will compensate for your investment. And then at some point as well, you basically you get the maximum return that you could possibly have. And it's that at that point where you want more or less to place your rotation age where you will harvest, at least from an economic point of view. So we run all these simulations. All these green lines, by the way, are all this variability I was talking about, all this uncertainty around the process. But let's not get into that. Let's see how these simulations translate into results. So I will first talk about the relationship between profitability and then these resilience indexes, this trait that Thomas was talking about, just to see how this play out. So in this graph, what I try to represent for just one site is on the y-axis we have one of the indexes, the resistance, and then in the x-axis we have resilience, and then lighter colors imply more profitability. And as you can see, the families that are more profitable are also the more resilient, or tend to be. So vigorous families are more resilient. So that's the first thing I wanted to mention. And now I'm going to summarize all the rest in this graph. So this is the effect of spacing. That's the kind of management alternative we used, genetics, and climate on profitability. Okay, so let's see if I succeed with this. So here in the y-axis, you have profitability in terms of LEV, the land expected value, in pounds per hectare. On the left-hand side of the graph, you have simulations for 2-meter spacing. On the right-hand side, you have simulation for 2.5-meter spacing. Then each block of 2 boxes is a climatic scenario, present scenario, mild scenario, severe scenario of climate change. And the red boxes are improved Sitka spruce. The blue boxes are non-improved Sitka spruce. Fine. So the first thing we see here very, very clearly is that with 2.5-meter spacing, you get much more profitability for all the scenarios, in any case. Now, don't jump into conclusions here. This is most likely an artifact of the simulator because we cannot consider mortality as yet. Therefore, it may well be that this 2-meter spacing is actually not realistic. Nobody's expecting at the end of a rotation to have 3,300 stems per hectare because there will be some mortality and perhaps some thinning. So this is just probably an artifact of the model. However, this is very clear. The more severe the scenario, the less profitable your forest is. And I don't think this is only for Sitka spruce. This is for everything, and this is very clear. And it confirms something that we already know. But if there's anybody who is skeptic, I think this is just yet another piece of evidence. And the other thing we see is that improved Sitka spruce works better than non-improved Sitka spruce in general, which, as you will expect, that's good news. However, not always. There's one scenario here for the 2.5-meter spacing, the more severe scenario, where this is reversed. And this is reversed for the 4 sites. So that's actually a quite important point. I will skip this one when I get to the conclusions of all this part. So as we have seen, there is a general loss of profitability due to climate change, quite an important thought to keep in mind. But second, it seems that the original selection for vigorous trees in the breeding program led to also more resilient trees. So this is like another success of the breeding program, I want to say. Now this is based on the hypothesis that the more resilient must be more profitable. However, this is just one trait we have tried. It's the trait of this growth response. It may well be that the trees that are not very conservative in growth, that really go for it, they may get into cavitation, and then mortality may kick in. So that's something that needs to be studied a bit further, because we come to this conclusion that we really have it, that our trees are really great. But still, that's quite promising results and very positive results. And then the last bit that is to be highlighted is that non-improved Sitka spruce is not to be disregarded as yet, because we have seen that very unanimously for all sites, it seems that under very tough conditions, it may be that Haida-Y QCI is actually going to do as well as improved Sitka spruce. So it's worth to keep this genetic variability just in case. The general conclusion of the whole project really is, to me, beyond the scientific fact, is that Sitka spruce is and should actually be used as a model to study resilience, at least for commercial conifers, because we know a lot about the genetics of it. So we can implement a number of things in here and then transfer the knowledge to other emerging species. And just one more minute before I go, this is like the deleted scenes in films. So things that didn't go well in the project, however, led to good stuff, to other byproducts that the community will be able to use very soon. So we had an interest to studying stem cracking as a proxy for economic resilience. Also because there is some evidence that there is a genetic control of this, and also we all of a sudden had quite a few micro drills. That's a device called resistograph, or people call it resistograph, and you can get a density profile of your tree very quickly. It's a bit coarse, but something really that allows you to sample quite a lot. So we didn't come to any conclusion with stem cracking. We don't know why stem cracking happens. But what we did while we were trying to do that was the first rigorous genetic-oriented stem crack survey in Great Britain. We also called it the first open source software to process the microdrill signal. That was our colleague Pierre Froment. And then we conducted also the first calibration of the microdrill signal for British Sitka spruce, so that this signal is transformed into density. That was Thomas who did it. And beyond that, I want to come back to this simulator, to this first stochastic growth simulator in Great Britain. This is the first time we do this, and any model can be included in this infrastructure. And as a result of it, you see now we are also doing other things, such as predicting strength grade in timber. So that's all on our side. Thank you very much for your attention, and we'll be very happy to answer questions later. Thanks.
Fantastic. That was really fascinating, Rubén and Thomas. I can see there's been quite a bit of discussion in the chat as well, which you might want to have a look at. Thank you for those of you who've posted questions. We will ask those after the last talk. I also just wanted to go back to Rubén's mention of Piero. Piero was one of the Centre for Forest Protection interns. We've been running internships for the last few years, and we'll be continuing into our phase 2. So do look out for those being advertised. And our interns all gave presentations in the first 2 of these webinars, and have really contributed hugely to the projects, as well as developing their own skills. I am now going to hand over to Annika Perry, who is a collaborator from the UK Centre for Ecology and Hydrology, who has been working with forest research on a Centre for Forest Protection project, looking at Scots pine and Dothistroma. So I'll hand over to Annika to tell you more about the project.
Thanks very much, Louise. Yeah, first of all, I just wanted to say a huge thank you for organising the webinar. I think it's been really, really helpful and really nice to see such a diverse range of research. And I think the whole programme has been really, really excellent. So yes, I'm going to talk mostly today about the work that I've done in trying to develop a simple and cheap method to predict host susceptibility. But I'm also going to give you a little bit of a background to the project in general, but a very, very small little bit of background. Richard does seem to be on the call. I'm not sure if he is right now. So he's the project lead at Forest Research, and yeah, a lot of this work has been what he's led. So I don't really want to speak to his part, but just to say that the project as a whole has been a lot more than what I'm just going to present here. So just to give a bit of context, Scots pine and Dothistroma needle blight, this is the species and the disease that I'm going to be talking about. So Scots pine has got this huge distribution right from the Atlantic to the Pacific and right from the Arctic Circle down to the Mediterranean. And the distribution that we've got in the UK, the sort of native distribution, is its westernmost part of the range. And the fact that it grows in such a vast area means that it's very, very phenotypically diverse. And globally, the most important disease of pine is Dothistroma needle blight, which is caused by a pathogen called Dothistroma septosporum. And it causes these very distinctive lesions on the needles. And when you get a lot of disease, as you can see in this middle picture here, it causes needle loss and growth retardation. And it can, in severe cases, also cause mortality. But the main reason that people are concerned is just really this sort of growth, the growth effect that it has as well. And so I'm sure it doesn't need pointing out to this audience, but understanding and predicting host susceptibility is crucially important to effectively manage both existing forests. In this case, I mean both the sort of native standing for populations, but also plantations as well, but also for planning the new forests that we're going to be planting. And so a bit of background for Scots pine and forestry. It's really, really handy actually having the background for Sitka already, because Sitka is by far and away the most well represented in terms of forestry. You can see that in terms of the stocked area and also the seedlings, Sitka sort of dominates, but Scots pine is the second most important species in the UK. There was a breeding program that sort of kicked off in the middle of the last century and involved domestic plantations and native pine woods. And there were selections and progeny test series. And then there was a reselection of parents from the general breeding population selected for height. Oh, sorry, I'm going a bit too fast, apparently. Apologies for that. Selected for height and straightness in 2002, and after this the program was closed. So but having said this, 50% of seedlings sold are still from seed orchards, so they're still a really, really important source of planting material. And you can see that in the last few years there's been an increase in plantings or in seedling production from Scots pine. And in order to understand why it is that Sitka is so preferentially chosen over Scots pine, you know, what drives these choices, I was involved in a project a few years ago that Richard was also in, and their part of the project was looking at what the stakeholder priority traits are in the UK and what drives decision making with regards to species choice for planting. And somewhat surprisingly, the biggest priority for both, well, overall for public and private stakeholders was disease tolerance. It kind of came well above climate resilience, but also, interestingly, tree form. In private stakeholders, tree form was more important, but overall disease tolerance. And there was this really seemed to be this need to have confidence that what was being planted would be tolerant to disease in the same way that people can rely on Sitka at the moment to be relatively robust against the threats. So as a result of this, this is the work that Richard's been leading in this project. We're aiming to sort of restart the Scots pine breeding program and build up those genetic resources and try to infuse what we understand about Dothistroma resilience or tolerance into this breeding population. And so this part of the work has involved taking two distinct sources for the genetic resources. So one is a native pinewood progeny provenance trial that we have, which I'll talk just about in a second. And these have been characterized for susceptibility, but also for growth and straightness and branching as well. And there are also the existing clone banks and seed orchards as well. And so from these genetic resources, the project has been working on performing controlled pollinations and also taking grafts in order to then have progeny trials ready to go, and also to develop a breeding arboretum with which to archive this material. So this is a really important strand, so very practical, that's being done and being led by Richard. And I mentioned the progeny provenance trials. So just a little bit of background about those. So this is a set of 3 trials that we have planted across Scotland. So there's one in the northwest of Scotland, one in the northeast, and then one down in the borders. And these are comprised of trees originating from 21 native pine woods. And they have this family structure as well. So there are 8 families per population. And they were established in 2007. The trees were planted out in 2012, and they've been regularly assessed for growth, for phenology, for susceptibility, and also for reproduction. You can see here, this was work that was done a few years ago on the bottom left. And some work was done looking to understand variation in susceptibility to Dothistroma, so DMB, Dothistroma needle blight, but also height. And working out what sort of gains could be made by reselecting particular families. So sort of coming back to the real fundamentals then, I've said that these trials have been assessed for susceptibility. And this is relatively straightforward to do when you're able to go back to the same trees time and time again. You've got the time to do that. Things are laid out in a nice sort of common garden progeny provenance setup. But in order to assess variation susceptibility, there's actually a few different ways that we can do this. So you can do an artificial inoculation, and this is inoculating plants in a controlled environment with selected pathotypes. And you do this for a defined period, and then you would then analyze everything. A natural inoculation is what I mentioned before, so that's what we've done with this progeny provenance trial. So this is planting, a common garden trial, so everything's exposed to more or less the same environment, and you know that there's going to be infection in the area, and then you track the progress. I should say, actually, the trial that I'm referring to, we didn't plant it thinking it was going to get infected, but by this point, Dothistroma seemed to be more or less everywhere, and so once we realised there was infection in the trial, we started assessing it. Another approach to assessing variation in susceptibility is just to survey what's there. So you can go out to forests, you can assess the standing trees, and you just record it. And then finally, there's the option of a detached needle bioassay. So this is where you inoculate individual needles and you maintain them in a controlled environment for a defined period of time. And you can see I've put ticks against the first 3 because those are ones I've been involved directly with. The first 2, and forest surveys are fairly routinely done. But detached needle bioassays are not something that we've done before as an approach to understand susceptibility to Dothistroma. So we wanted to really see if we could develop this as a tool. So when comparing the different methods, you can see here that there's, sorry, it's not done the animation as I wanted it to, but anyway, I think it's fine. You can see that if we just kind of simplify everything, there's a lot of differences in terms of the experimental design and in terms of the practicalities here. But if we just take 2 of them, we've got host variation. You can control the host variation that you're analysing in artificial and in a natural inoculations. You really can't in a forest survey. It's just whatever happens to be planted there. But in detached needle bioassays, again, you can kind of select which of the trees are going to be included. And when it comes to practicalities, a really important point is the complexity. And with complexity, I'm kind of combining lots of things. So I'm talking about how expensive it is, how much time it takes, how much space you need. And artificial inoculations and natural inoculations both require a lot of time, resources, and they tend to be fairly expensive because you need to have both of those in ready supply. In comparison, forest surveys are relatively inexpensive and simple. You just turn up and you do your assessment. And similarly, detached needle bioassays, they're relatively simple. You have to have a bit of space for them, but you just need some bench space. You don't need to plant them out in it and let things grow and grow. So some of the questions that we had coming into this then were, how well do bioassays reflect susceptibility in the field? Also, can bioassays be used to identify susceptible and resistant trees? And also, how consistent are the results from the same tree or from the same clone, if we're talking about grafts, sampled at different spatial scales, so that we can get a feeling for actually how useful the method is. So yeah, my research question. So first of all, then I was asking, how well do bioassays reflect susceptibility in the field? And so for this, I used this common garden progeny provenance trial, which has been surveyed for susceptibility from, well, so near enough 10 years now. And I'm not going to go into detail here, although I'm very happy to explain about it. But each year, with a couple of gaps, I've been assessing every single tree in the trial. And we've got a nice distribution of trees ranging from sort of high to low susceptibility. And for those, I've got a sort of an individual susceptibility value for each tree. And I've also assigned them based on the sort of tail ends of the distribution. Some are assigned as resistant and some are assigned as susceptible based on their values. And so these trees were then used as the source for bioassays. And the sort of hoped outcome is some sort of validation. So bioassay susceptibility should correlate nicely with field susceptibility. That's the hope. So my second question was, how good are bioassays at identifying susceptible and resistant trees? Now, in this case, I realized that it's susceptible and resistant is can be quite subjective. It can be related to the environment. So like I say, I've defined in this environment which trees are susceptible, which are resistant, and I want to see if the bioassay picks up those same patterns. So the same trial again, put into bioassays, and in this case, the susceptibility in the bioassay, you look at the distribution, you assign some of these as resistant, some susceptible. We've got a cutoff of about sort of 15% tail ends of the distribution to try and kind of capture just the extremes of the trait variation. And then assigning these groups, how well does these groups then correlate with field susceptibility, and also can trees which are identified as resistant in the bioassay correspond with those which are resistant in the field as well. And hopefully we've got a sort of accuracy outcome from this. And finally, I would like to know how consistent results are from the same tree sampled at different spatial scales. So for this, I considered sampling within a single tree, so within different positions in the crown. Sort of a moderate spatial scale, so within a single site, but ramets. So ramets are the same genotype, the same individual tree, which is then grafted onto different rootstock and then planted and sort of grows into different trees. So ramets should be genetically identical individuals. So when you've got multiple ramets in the same site, how do they look in the bioassay? And also large scale, so the same clones that are planted at different orchards. So these final two, the sort of moderate and large scale, these come from trees which are planted in seed orchards, whereas this small scale comes from the common garden trial that I mentioned before. And these are all popped into a bioassay. And then again, I want to look and be able to compare results for this to be able to assess some sort of reliability score as well. So I've been talking a lot about the detached needle bioassay, but what do I actually mean by this? So I've tried quite a few different approaches, and this is what I've ended up with. So you start by collecting your sample from a tree, you store it in just a poly bag and keep it at ambient temperature, and then when you get back to the site, you pop it in a fridge, keep it chilled. And then when you're ready, you take individual needles from each branch and you pop them in a specified location. And you repeat this. So this is one block and this contains one needle pair from every single tree. And you might repeat this 10 times so that you've got 10 replicates of the same setup, just randomized each time. And keeping the needles wet, because really we want to make sure that we're especially in the very beginning stage, we wanted to make sure that it was sort of replicating a sort of, you know, what might happen if you're exposed to or susceptible to Dothistroma, and that generally comes along with having a lot of needle wetness, because that's conducive to pathogen growth, dispersal and development as well. And then what I was doing was assessing the brownness. So brownness is very, very simply just what percentage of each needle pair is brown at each point. So this is 21 days after the start of the experiment. And then again, I did another measure at 49 days after the experiment, so about 7 weeks. And then quite simply, that was it. I just look at brownness as it progresses over the course of the period of about 7 weeks. And then there's take the data and then perform these steps that I was talking about before. So validation and accuracy, and actually see what we can get from it. So you might be wondering why I've not mentioned at all about inoculating the needles with Dothistroma spores. Why wouldn't I do this? Well, I did. I've done a lot of inoculating detached needle bioassays with Dothistroma spores, and I was constantly coming up with issues because of pre-infection. So Dothistroma is more or less everywhere in the landscape, and I tried many, many different things to try and sort of prevent or minimize the risk of pre-infection. I sampled at different points in the year. We even tried isolating the needles in the bags that are used for the controlled pollinations to try and stop the spores from coming in, and it was just, yeah, it was just not working, and it means it's very hard to be able to disentangle the results from pre-infection with those from inoculation. So I also found needle brownness, to be honest, was a sort of and I measured it at first just because I noticed that some were turning very brown and some just do not turn brown at all. They can be in a wet box for 7 weeks and they remain totally green by the end. And there were other added benefits as well. It's much, much quicker. Just the scoring is very, very simple. It's cheaper. You don't have to then start buying consumables and reagents to do an inoculation, and it's safer. I wasn't chopping, you know, cultures out with scalpels and things like that. So overall, I was thrilled that needle brownness was, you know, a much, much more effective way of trying to understand susceptibility in a bioassay environment than inoculating. It was a good result. So for my first question, I was asking, how well do bioassays reflect susceptibility in the field? So just for a little bit of context, this is, you know, if you look over a year, then where I've put orange, this is where there's really not much in the way of growth. You might get a little bit at the tail ends here, depending on how warm it is. But generally speaking, bud burst progresses from about April, and you get, it varies quite a lot, but you get it continuing. So you get the full needle emergence and elongation by about June. And then you get active growth happening over this period, and then it slows down again by the time you get to sort of October time, again, slightly depending on the weather, on the climate. And so I did bioassays where I collected needles in July, in August, and in October. As I said, at first I was trying really to minimize the chance of pre-infection. So I thought that sampling earlier would actually be much better. But you can see there's really no relationship at all if you look at bioassay susceptibility compared to field susceptibility. And in fact, you can see that bioassay susceptibility, most of the trees were really highly susceptible. And this is because the needles were so brand new, newly emerged, they just succumbed to the bioassay conditions really quickly and all turned brown very, very early. In August, we see a slightly better pattern, sort of a tentative positive association, but a huge amount of noise. But by August, we see actually quite a nice, clear, again, there's still quite a lot of noise, but, you know, that's fairly standard. But we at least see a really highly significant positive correlation between bioassay susceptibility and field susceptibility. So I was really pleased with that. Validation, tick. So my next question was, how good are bioassays at identifying susceptible and resistant trees? And similarly, I just used the same approach over these 3 time points. And you can see that actually in the bioassay, trees which were assigned as susceptible do have overall higher field susceptibility, but the difference isn't significant until we get to October, and then you see really, really highly significant differences. So trees which are assessed as classified as susceptible based on their bioassay do correspond with significantly higher field susceptibility values as well. And you can also see that the accuracy, which, I mean, we can pretty much ignore July and August, seeing as they're not useful. You can see that the accuracy is actually higher for susceptible trees. And you can see that here, the box plot here is much smaller. It seems to be much more accurate to define susceptible trees compared to resistant. And the error rate is much lower as well for susceptible trees. So it might mean that this method is actually particularly useful for identifying susceptible trees. And so my next question was, how consistent are results from the same tree sampled at different spatial scales? So first of all, there was a small scale. So this is branches sampled from high up in the canopy, in the middle of the canopy, and low in the canopy. And I should say these trees are now about 17 or 18 years old. They're generally sort of between about 4 and 6 meters. And you can see that there is no significant difference among any of these, whether you sample in bioassay susceptibility, regardless of where you sample them. And you can see that they're very highly correlated as well. Susceptibility is for trees, for needles sampled in the bottom of the crown, is highly correlated with susceptibility for needles sampled in the middle of the crown. Slightly less between the bottom and the top. But what you find is if you then just look at those trees which are assigned as resistant or susceptible based on their field susceptibility, you see actually there is only a significant difference if you look at needles which are sampled in the middle of the canopy. And I think possibly the reason for this is that because these trees are starting to drop their lower branches, I think there might be something to do with needle condition in these bottom branches, which just makes them less reliable. Similarly, in the top of the canopy, you've got exposure to a whole range of more extreme environments in terms of the abiotic and biotic environment, or particularly the abiotic environment. So again, it might be that crown position is associated with needle condition, and in the more benign environment in the middle of the canopy, that's where it becomes easier to pick apart those differences. It's possible that you see a reduction in this effect once canopies close, but that's something I need to look into. So there's sort of a tick for reliability, but, you know, I think a little bit of cautiousness there. And then I looked at moderate and large scale spatial scales. And so you can see that for the moderate spatial scale, so this is looking at the difference among ramets of the same clone, you can see that there isn't a significant difference. So it suggests that using a single ramet per site may actually represent a clone susceptibility at that site. The caveat being that orchards show highly significant differences. So you can see here, these are 3 of the orchards. And although, you know, quite often the trend is in the same direction, I couldn't do a proper genotype by environment interaction here because not all of the genotypes were present in all of the environments. But you can see that there are some interactions, but generally speaking, things move in the same direction. But clearly there is a big effect of site here as well. And it's probably just the case that trees are growing for a really long period in a particular environment, and that has big effects on the needles as well. So I would say apply caution to the use of trees from different environments. So there's sort of a plus point, you know, it's really good that you can use, you know, within sort of moderate spatial scales, there isn't a big effect, but I think just be very careful about inferring things from trees grown in different environments. So some of the recommendations based on this work, I should say this is just like a little part of the work. I've tested it in other situations as well. But the recommendations sort of for the basics are to collect later in the growing season if possible. We think this has something to do with sort of robustness of the needles. Avoid directly comparing susceptibility of trees which have been grown in different environments, as I said. Apply a consistent sampling methodology, so make sure you're sampling from the same location within the crown. Include as many trees as possible in each bioassay. I thought at first if you just, I could just include a very small number, but actually you need that a scale in order to be able to pull apart those tail ends. I just think it's not so useful as a technique otherwise. And be really cautious, much more cautious when classifying trees as resistant as compared to susceptible. So the next steps, I'm going to repeat the bioassays using the same trees and the same time of year, just to make sure that we've got some measure of reproducibility. I'm also interested in using younger seedlings because it's possible there's an ontogenetic response, which means that it's a response based on the age or its stage of development. And really for this approach to be useful, we also want to be able to use it to assess susceptibility in very young seedlings. I'm really interested in comparing artificial inoculations alongside the bioassays to compare the response, and also to understand the mechanisms underlying this response. And a little bit more about this, I think what we are potentially seeing here, one of the hypotheses is that the response we're seeing isn't a sort of a direct, it's not directly saying that this exact same mechanism is at play. It's possible that the bioassay is picking up, it's a sort of a proxy for susceptibility because there might be similar sort of needle structure. There is something to do with the needles which responds in a similar way to the conditions of the bioassay and kind of is able to slow down that browning response, which is also something that we see in the response of susceptibility. So I'm really keen to understand, and actually we've got a phase 2 CFP project, and part of this is going to be doing a little bit more work on looking at the sort of mechanistic processes underlying this susceptibility response as well. And then also what I'm keen to understand is what's the optimal timing for this? So as I said, brown has started off as a bit of a, oh, that's interesting. So I hadn't really thought about the best timing to do. And so I think if I can conduct weekly surveys, I could then optimize this process further. So just to say that there's a lot of people involved in various aspects of this work. The list's here. Massive thanks, obviously, to Richard as well. Yeah, thank you all for listening as well. And I'm very sorry for racing through things at the beginning. I was very aware of time and I was trying not to fall behind.
Fantastic. Thank you so much, Annika. And I know that represents a lot of trial and error with optimizing the methodology for it as well. And as Annika said, some of this work is going to be continuing in phase 2. Right, we have a little bit of time now for some questions. I'm probably not going to get through all of the questions in the chat. So speakers, if you can perhaps have a look at them at some point afterwards. I think the chat will be available for everybody as well. Sorry, I'm just adding a spotlight so that people can see our speakers. That's great. Okay, so first question for Jaz. This is from Rory Lunny, did any host patterns that were linked to the native range show a greater incidence of threat if they were coastal or interior? I'm not sure if that's something you looked at.
We didn't look at geographic range. We looked at host range in terms of the range of hosts that threats are found on, but not actually a geographic range. There is something that we could be added into the modelling. It was a consideration, and maybe for future work, it'd be good to include sort of geographic components. But this is more of just a general anywhere in the UK for any given ideally any given species, member of that species.
Yeah, thanks, Jaz. Annika, the next question's from you for Jaz. I don't know if you want to ask ask that?
Sure. I'm just going to scroll back because I can't remember exactly what I said. But yeah, it was just to say, while I find it, I was really, really interested in it because we've got a project at the moment that is looking at risks to of planting non-native species. So sorry, let me find it. And so yeah, the talk was very very timely and very interesting. And I was just wondering if you know how well the approach will work to predict the interactions which aren't particularly damaging in their native range. So there's lots of examples where, you know, stuff sort of seems to be ticking along fairly benignly, and you pop a new host, a naive host in, and then it explodes. And so how well is the approach that you were outlining able to pick up those interactions as well?
Well, first of all, you know, it's if there is information on that threat being present on that host. So, I mean, there's going to be a little bit of a bias in the data. Things that are more severe probably get noticed more and appear in the data sets. But, you know, if it is not particularly damaging, but is noticed to live on that plant, be hosted by that plant, then the modeling should hold. But also we did run sort of an additional analysis, and turn up in the report and stuff when it finally comes out, about severity. We found a sort of severity data set, and we ran a correlation or an analysis to check whether there's a correlation between the strength of the prediction, so the likelihoods, and the severity of the threat, both the severity of the interaction of that threat and also the severity that threat normally has in just other plants. So sort of it was seeing if there was a correlation in either direction or both. And what we found is that there's no correlation between like the damage that is done and the strength of the likelihood that's predicted. And in a way, that's probably a good thing, because that means that, you know, we're not just predicting things that are known to be very damaging. Basically, we're just predicting that there's the likelihood that it will be on that plant, and then yes, it means it warrants further work to identify whether or not it's going to be severe. But it should mean that things that aren't too severe on their native hosts aren't being overlooked. There's no like negative correlation there.
Yeah, thank you, Jaz. And then the next question is whether you've extended the model to model pests that are in Europe that are likely to come here in a changing climate.
It was not. It was just UK native, but it would be very easy to, you know, rerun this with an extended data set. That's the sort of the intent of this, is not to be a one and done. It was more proof of concept. Look, this methodology works. Hopefully now, any data set you wanted, you could say, you know, Europe native plant threats, and then non-European, non-natives, and yeah, you should be able to extend this basically as much as you wanted to predict.
So this
for
is the initial model was UK organisms, but could be extended. That was from Jonathan Burton, who's also suggested you could look at the International Plant Sentinel Network planting schemes to check the model predictions. So if you have a look in the chat, there's a link to that. Rubén and Thomas, there's a little bit of discussion about your talk, and Richard has answered some of the questions, so you might want to have a look at that. One question on the economic graphs from John Burgess says, do they take account that larger saw logs have a higher value? So retaining a stand for longer brings relatively higher value timber.
Yeah, they take into account that we obtain timber prices for historical timber prices for different sizes and everything. It does take that into account.
Yeah, so that is included in the model. And a question from Louise saying, for context, how much of the Sitka growing in Britain is improved Sitka?
I think Richard answered that one.
Has he answered that?
Yeah. Okay. I think he said 95% of what is planted is at least from seed orchards. So, yeah. but
So
currently
actually
most of it.
A huge amount. Okay, and a question for Annika. It's great. This is from Alex Steep. It's great to hear that Scots pine is being looked at again for a breeding programme. It's got a strong symbiotic association with mycorrhizae, and would this be accounted for when measuring gains in a breeding programme? Are all the mycorrhizal associations equal? Mycorrhizae have been shown to improve pest resistance or resilience to other tree species. So would this be the case with Dothistroma?
I can't speak for mycorrhizae. Perhaps Richard can, pop something in the chat about whether it would be able to be looked at for gains. I suspect that the vast majority of the differences in mycorrhizal associations will be due to site rather than due to genotype. I know that some work was done in the past, and I don't think it was a very big effect, but I could be wrong on that with Scots pine. We do have a project looking at the microbiome, which for Dothistroma certainly is more relevant because it's a foliar pathogen. You might find that if there are root pathogens there, the mycorrhizae are, particularly important in that interaction. So with the microbiome, we're interested in whether the host genotype and susceptibility interact with the microbiome as well. But in a similar kind of a way, is that it might mediate that interaction. But yeah, no, it's an interesting point. I'm not sure.
Brilliant. Thank you. And Rich has just added a lot of older stands of Sitka will be unimproved, but since 2000 it's 95%. And he's also added something to that last question, Annika, saying you could consider incorporating treatment into planned progeny tests under the next project, and they're looking for sites to plant in 2027, 2028. So do get in touch with Richard Whittet if you think you have a suitable site. Right, we're nearly at 11:45. I'm going to leave it there. I want to say a massive thank you to our four speakers today, and also thank you to Katie, one of our interns, who's been helping, just making sure that everybody's let in, keeping an eye on the chat. So thank you very much, Katie. 3 of our interns have been helping with these webinars, and they are all finishing their projects this week, so big thanks to them. And I also wanted to say a very big thank you to our funders at DEFRA, who have been funding the Centre for Forest Protection and encouraging all of the collaborations that are happening between Kew and Forest Research, and then with other collaborations. And have been looking at it. We're now expanding the scope of the research programme. And as I say, the focus is really on these new innovative methods that you've heard about today, which is giving our scientists the opportunity to perhaps be a little bit more experimental in trying out new methodologies to look at the ways we can protect our future trees and woodlands. Do take a look at our website and sign up for our newsletter. There will be a number of papers, reports, and report summaries and trade journal articles coming out over the next few months, as well as information about the new research projects that are starting for phase 2 in April. I'd like to thank all of you who've attended for coming along and listening to our scientists and taking such an interest in the work that's going on. So thanks very much, everybody. I'm going to end the meeting there.